Hello, everybody. My name is Banu Priya Sridharan. I am a postdoc at the Scripps Research Institute in Florida. And today I'm going to talk about addressing the scalability of human iPSC-derived neurons for HTS or high-throughput screening implementation using phenotypic screening approach. A brief overview of the presentation. Initially, I'll talk a few minutes about the Scripps Research Institute's the screening center over here, and what is the background and the motivation behind this project, and how we went about establishing an assay for this phenotypic screen, and also kind of briefly talk about our disease model using the induced neurons for the Syngapi disease, and also quickly summarize and tell you a few things about where we are headed next with this project. So briefly, the screening center here at Scripps started in 2005, and it's an interdisciplinary team made up of biologists, chemists, and engineers. And we have industrial scale robotics, which is what makes us very unique, being an academic lab in an industrial onset of equipments and liquid platform dispensers and everything, including a huge drug library. And the funding is driven by both NIH, federal grants, and from the pharma and biotech. And there are you could divide the lab into three parts. The first part is where I work, or the fun part, as I like to call it, where most of the assay development, where we get assays on 96 well plates or 24 well plates, and we optimize it to 384 or 1536. And then the second part is the robotic part, where it goes into the robot, and our robotic engineers overlook this screening part with 1536, and we go up to 200,000 samples per day, and everything is automated at this point. And the last part is the compound management that comes more into play once the assay is past the primary and secondary screen. And the tools used for this expedition pretty much majorly comes from Thermo Fisher. Starts off with the VitaPower, using the Cell Insight. We needed the BSL-2 facility, UV transilluminator, Perkin-Elmer's Envision, Pintold Station, and Thermo's well-made. Why do we care about induced neurons, right? So there's a huge lack of predictive preclinical models, and the golden standard today for neuro drug testing is pretty much just mouse primary neurons. But they don't obviously translate to human neurons because of the increased complexity. And also, in talking about human neurons, right now they are being made from iPSCs, but they're not being made in amounts that are scalable for industrial scale screening. Also, they're very slow to make human neurons, and it's very variable from batch to batch, from patient to patient. Now, how do I make these human neurons, right? So the rationale behind induced neurons from iPSC being there's a virtually unlimited supply of iPSCs from patients, and there's no ethical complications because these are adult cells, and you could also make PDX models and test both the 2D and upcoming experiments using 3D, and also bank it and go back to any further testing you want to do with these iPSC-based neurons. And using human neurons, phenotypic assay development is also another area that's gaining a lot of momentum, and we want to develop assays that are statistically significant using good plate statistics like Z primes and signal basal, and have the best assay in the shortest time possible. So the current techniques to make induced neurons are, please focus your attention on the left side of the slide. As you can see, it's numbered, so it goes from fibroblasts to neurons, or going from fibroblasts to iPSCs, from iPSCs to neuroprogenitor cells, and from neuroprogenitor cells, or NPCs, to induced neurons. Now, obviously, these are all long and complicated multi-step approaches. So what we wanted to do was to get an iPSC cell line with the ability to develop a fast, cost-effective, homogeneous method to make induced neurons and develop a human phenotypic assay that are high-throughput screening friendly. How do we go about doing that? The first step is to get a iPSC cell line that was validated from Thermo. This was received as a gift from Thermo. And we were able to bank, because it's a cell line, we were able to bank about 100 cryovials with early passages, and we routinely QC'd it for mycoplasma and pluripotency from time to time. And also we have an RFID encoded freezer pro to have a good record on how many vials of each passage was remaining. Now, these are all very important when you talk about large-scale robotics and screening and how to maintain these systems. In pluripotency was a two-step confirmation in our case. So the image on the left was the first immunocytostaining of the Cas9 iPSC. And I don't have to preach to the choir, but you all know SSEA and OCT4 are surface markers for pluripotent stem cells. And then these cells checked out positive for these both markers. And then we moved on to a more comprehensive and detailed TACMAN scorecard. Once again, that was from Thermo. So what this scorecard does is a PCR-based method where we put in the RNA or the cDNA, and it yields out, it already has primers in a pre-plated format, and once it kind of queries it against the database, it gives you a scorecard that has all the information like what are the self-renewal genes, and in the form of an algorithm score, and does it fall within the ectoderm, mesoderm, or the other three germ layers, basically. So as you can see with the right arrow pointed, it falls under the self-renewal box, thus confirming that these cell lines are indeed pluripotent, and they do not fall under any of those germ layer, which is this is what we needed to confirm. So once these two QCs were in place from time to time for every passage, we kind of moved on to actually start making the neurons. So these neurons were made by plating the iPSCs, and I won't go into too much detail, but briefly we used lentiviral transduction and DOX activation and puramycin selection to make induced neurons from iPSCs. Now, once they were selected using puramycin, these induced neurons were plated, or INTs, I'm just going to call them INTs. They were plated on 384 well plate for now, and we were able to pin them with compounds and then analyze them 48 hours later. So our goal in this case was to just reduce the timeline, because most of the neuro-based assays were at least 14 days and up. So we wanted to kind of shorten the timeline and say we could do this neuro screening within 9 or 10 days, right from plating the iPSC to the final readout. So the cells is a check mark. We were able to get a lot of cells on demand. Now, next step to make these lentivirus was to get a lot of vectors. So we got a bacterial stap from AdGene. All of the sequences are public. Here is the AdGene ID number. We were able to make transform using STBL3 cells. And once again, I won't go into a lot of detail on how we got these plasmids, but it's pretty straightforward. And the only thing we had to do, because it's the HTS lab, was to use MaxiPrep and create a lot of plasmids. And as you can see, we confirmed the right sequence of these plasmids using restriction enzymes, and all those lists is also on the right side of the image. Now that the sequences are verified and we have the right plasmid in place, we once again went to the next step, which is to make the virus and titrate it. For this process, once again, we used Thermo's ViraPower system, and using 293 FTs, the plasmids were expressed and they were extracted and titrated. So now the image on the right, you see a paneled GFP. Using GFP indicator, we were able to get good expression at MOI of 1, which is great. And now we have a method to make bulk lentiviral vector so that you can make neurons on demand. So neurons are very sensitive cells, and they need a glial layer for support when they grow, especially for long time points. So then we had to explore everything using the height throughput in mind, right? Scalability in mind. Now, how do we use human versus animal astrocytes, or could we explore other surfaces? So let's go to the first option, which is to use glia. So we did first think about using human astrocytes because these are human neurons. But what happened was that these astrocytes required Geltrix-coated surfaces for growth, and we realized that these human astrocytes do not proliferate. Once again, this is not a scalable or an affordable proposition. Moved on quickly to Sprague-Dawley rat astrocytes, and the problem here was we were able to get a lot of astrocytes, but it took about 4 passages for cleanup because the primary neurons have a lot of other type of cell microglia contamination, so I had to use RSE to clean out the cultures and get a pure population of astrocytes. So it's not only time consuming, but it's also reagent heavy. All right, so what did we do next? Alternative solutions. We explored something called the surface coating. As most of the neuroscientists already know, poly-D-lysine is a wonderful substrate that helps these neurons to grow. And our lab has already published a paper where we used poly-D-lysine-coated 384 well plates for a full primary screen for mouse primary neurons. But I was able to validate them on human primary neurons. As you can see, the top right panel, these were neurons imaged at day 23 on a 384 well plate that had poly-D-lysine. So these are already pre-coated. We just get them as well plates, which makes scalability easier. Now, the second approach instead of D-lysine is ornithine. So we followed Kristen Bernan's lab protocol to manually coat poly-D-lysine and laminin on a clear 384 well plate. And once again, as you can see, just within day 7, there was a lot of proliferation and a lot of neuro branch points in these neurons. So the question is no longer glia, right? So now that we're able to get good, healthy neurons growing, it now changed to what is the best surface coating, which is a scalable question to be able to answer. So once we got all that figured out and optimized, the last step was to actually figure out what kind of endpoint or analysis would make a lot of sense for these induced neurons. And using appropriate controls, do you want to track positive and negative control? We use DMSO or Bartholdi or rotenone, and also look at Neurite Outgrowth assay as a starting point using CRC of the control compound, the 2-channel assay, which I'll talk about very soon. Neurite Outgrowth assay, once again, we use Thermo Fisher's high-content reader to look at these Neurite Outgrowth. And the reason we went for the Neurite Outgrowth was because it's a well-established endpoint for neurotoxicity. A lot of labs use them, and it's early discovery, and you could always multiplex a Neurite Outgrowth with other readouts like MITRE-tox. And the output parameter, which we were most interested in seeing, is the cell body or the neurite count, neurite size, and neurite length. We also were interested in looking at branch point count and to see cross point count as well. Now, the nucleus count, since I said it's a 2-channel assay, right, the DAPI channel or the nucleus channel was able to provide that information. And the other neuron-based parameters such as neurite, cell body, and branch point comes in from the 2nd channel, the FITC or the rhodamine. It really depends on what dye you're using. In this case, as you can see in the panel below, called sample analysis, if that is a neuron, and this is an image directly from the manual from Thermo's high-content reader, and that's how the segmentation and nucleus, cell body, branch points are all counted. And I have a little image here that shows little green, blue, and yellow colors on the same image that shows how these images are actually being segmented and counted. Now, the next goal, so to speak, is to adapt this into a 384 well plate and to actually produce high volumes of INTs tested on various treatments, ascertain rapid readouts, and also kind of validate plate-to-plate, day-to-day, and well-to-well robustness for high-throughput screening. So here is a short 384 well plate flowchart, how this is done. The, hold on a second, the 384 well plate starts off with 2,500 cells in a 30 microliter culture medium. This is per well. And after an overnight incubation, the compounds are added via Pinto, and in this case, we're using 100 nanoliter Pinto. And after 48 hours of incubation with the drug, these cells are then fixed. They are stained using, once again, Thermo's Neurite Outgrowth kit, but any other Neurite Outgrowth kit should work as well. And then they are also read in 3 channels using the high content reader. Now, the data analysis as such uses only 2 channels, and the output, as I already mentioned before, are the valid neuronal count, the Neurite length per well, and Neurite count per well for now. And the final volume of the assay is 30 microliter, and the final drug screening concentration is 8.3 micromolar. And currently, we were able to do low-pack screen on these neurons, but this is the Neurite outgrowth layout, so to speak. So please focus your attention on the first or the top panel. So if you look at a 384 well plate, right, it has 24 columns and 16 rows. The first 2 columns, column 1 and column 2, that says no cells because we wanted a column of blank for any background subtraction because it's a high content assay. That's a standard that's being followed. The second column is a DMSO treated or the positive control that does not affect Neurite outgrowth at all. And on the last 23 and 24, 24 is the Brafaldin A, that is a negative control in this case, or a compound that causes the Neurite length to shrink. So that's a negative control we used. So the inhibition is high for these compounds. So it's an inhibition assay. And as you can see in the chart below, the percentage response refers to percentage inhibition. So if it's a compound that kills the neurons or kills the neurite, you would see a high rate of inhibition, which is what you see in the Brafaldin column. These dotted points refer to Brafaldin A-treated cells, whereas the columns in the bottom, which is close to 0, which means the neurons are fine and healthy, are all, they all represent the baseline of the 0. The reason we have this dotted format is to look at if there are any dispense issues or if there are any plating issues or anything like that. Such a format, and if you look at the small tab that says plate stats, you look at the percentage CV, and a signal to noise should be about 10-fold in this case, and the Z primes are the ones we most care about. And here you go, I have written down the Z prime of 0.5. In this case, is considered good for further analysis, and that's a standard that's commonly being followed for passing high-content screening applications. Now, what that actually translates into images is what this slide shows here. The left top panel shows an example of how a DMSO plate looks, and if you see, you can see a lot of neurites happily growing everywhere, and if you see Brafaldin A, a 10 micromolar final pinning concentration, you would see that there's almost very little nuclei and neurites remaining, and that the panel right below it shows how the software is able to isolate these and give us a readout in terms of, once again, if you look at the panel on the right, look at how does that translate to a neuronal count, how that translates into a branch point, neurite count, and most importantly, neurite length. So I guess the conclusion of this slide is that we will be able to use this Neurite outgrowth assay as an endpoint, especially to validate any compounds that cause neurotoxicity. Beyond going just for high throughput applications, we wanted to also see if these neurons are actually neuron-like. So we collaborate with Gavin Rumba, who is an investigator or a neurobiologist at Scripps. And his lab, we were able to collaborate and kind of test out that by MAP2 and synaptophysin staining, we were able to check that at day 30, these neurons definitely stained for all mature markers for developmental neurons like dendrites and synapses. And we also have data, which is not here, but we have a 30-day electrophysiology data that shows these neurons firing signals at 30 and 60-day time points. Now that we were able to establish that the neurons are scalable and they are indeed mature neurons, and they are screenable in a phenotypic fashion. I just wanted to show you what that means in terms of cost, right? So going from a flask of Cas9 hiPSC to go to neurons, and by puromycin selection, going to induce neurons. And of course, in this case, I only used high content readout, but there are other readouts like MitoTox or Envision or fluorescence-based quantitative readouts. But if you want to do something like that, one plate of iPSC alone would cost about $20, or for a low-pack screen in triplicate, it would cost about $300. But if you were to induce neurons, it's about $2,000, which is not bad. It is very scalable compared to using mice primary neurons. Now you have a chance to actually use human-induced neurons. And moving a little bit ahead, beyond just this project, we have been fortunate to collaborate with Gavin Rumba's lab, who works exclusively on Syngapi disease models. Now, Syngapi is an intellectual disability that causes autism and epileptic encephalopathy, and the Syngapi1 gene is haploid insufficient. So currently, we were able to generate using CRISPR, and once again, this is a Cas9 iPSC cell line, so it was easy for us to create a Cas9 Syngapi iPSC. We were able to use that Cas9 part of it, and using CRISPR-Max, make Syngapi iPSC. And once you have a Syngapi iPSC, it is now, through the same method that I just described, using all the lentivirus, and make Syngapi-induced neuron out of these iPSCs. Now, our next step is we already created Syngapi neurons, but we're now going to compare homozygous and heterozygous knockouts of these alleles, so that now we can use it as a control when we screen patient iPSCs or patient-induced neurons. The cell lines are now being validated by NGS. And this is really beyond just neuro, right? So the future directions, at least from our lab, is to, first of all, harden the existing system. There are so many options with respect to coding and plasticware, and to kind of get the best what is out there. We have to spend a lot more time validating on different equipments, validating on different plate types and vendors available, and also to optimize a virus titer in-house, and work on looking at different seeding densities depending on the length of the assay and depending on the endpoint. That kind of leads to my second point, is to broaden beyond just the high content that we just used for Neurite Outgrowth, also go to other mitochondrial health and other type of endpoints for neurons. And the third, which we already underway, is to look at Syngapi, like we just said, but also kind of talk about brain on a dish, because our lab has expertise on 3D organoid model, so we want to exploit that expertise and work with 3D-induced neuron models to look at early developmental disorders. And also just go beyond just neurons, like, for example, ocular degeneration and hepatocellular diseases, mainly because we have other needs because we have an iPSC cell line that can virtually be made into any cell line that we want. So these are very broad what we want to do for the next kind of approaches. None of this work would have been possible without this fantastic team. So I would like to thank Louis and Tim, who are the lead ID or the screening center directors. And for assay development, I want to thank Shurong, Vernie, and Deanna for helping me with high content and for navigating this lab as such. Fakar and Pierre have been real help as far as robotics is concerned. And also we want to thank Gavin, Chris, Murat, and Nerea, all from Gavin's lab, who have helped us with cytostaining and for 30-day electrophysiology data and other neurobiology endpoints. And we also want to thank Thermo Fisher, especially David, Mark, Eric, and Stephen for helping with expertise or any questions we have, and also for providing some reagents, and also checking back and helping us whenever we had some questions about how to use these cell lines and these reagents. And also we want to thank NIH for providing us the funding to be able to continue doing this great project. Well, thank you so much for your attention. I have my contact info on the first slide, so if you have any questions, feel free to email me. Thank you.